from Cornell. She is a viticulture professor there. She works with the grape and wine industry in New York, and also teaches classes at the university. Has a really interesting background. One thing I'll say is that she was the chair for Dr. Justin Shiner during his PhD time at Cornell. And so she's got a little bit of Texas experience there, haven't she? And I think she did a great job. So we're excited to have her here to talk about her research. And also she's been able to explore Texas and our industry and talk with our colleagues, especially in extension. So we're excited about some future collaboration with her. But Dr. Vanden Heuval, thank you for being here.
Thank you, Michael. Thank you, everyone. I'm glad to be here. So today I'm going to talk about some work that we've been doing on leveraging remote sensing to help guide sampling. So at first, a little bit about Cornell, in case you don't know us. So we're located in Ithaca, New York, and then we have an agriculture campus that's in Geneva, New York, about an hour away. We conduct research, teaching, extension in lots of different topics, but particularly agriculture. So we are actually the only Ivy League university that has an agriculture program. And we specifically have a viticulture and enology major that we started in 2008. We have a winery on campus. We have vineyards on campus. Our students make wines and study science through the lens of viticulture and enology. As Michael mentioned, this is where Dr. Shiner did his PhD, where he did at least spend some time researching whether his cowboy boots with no treads really worked on ice or not. And he can tell you the results of that research. So, yeah, he's got the scar on his head for it, yeah. So my research focuses in two areas, but what I hope to try to convince you is that this is really all one area. So on the left here, I have sustainable soil and vine management. So I do lots of studies where we are starting integrating animals into vineyards as a key to sustainability, nutrient cycling, and soil organic carbon storage. I work on mycorrhizae colonization of roots and whether we can inoculate grapevines to make them more resilient in the face of climate change. And I work on some disease models and how we can improve those so that our growers are more likely to use them rather than just doing calendar sprays for some of our diseases that we get in the Northeast. Over on the right-hand side, I have digital agriculture. So in 2018, I think it was, Cornell started a Cornell Institute of Digital Agriculture and really initiated a ton of research where engineers work together with plant scientists. And this is where a lot of my research program has focused. So I've got some examples up here. The one in the middle is that we designed a simple computer vision method for counting clusters. Because before this, our growers would estimate their yield by looking at their vines and saying, that looks like 6 tons to me, right? So now we're working on berry counting as well. Top right is actually a newer model we've designed. We call that Grape SAM because we started with the segment anything model. So we can just take regular vineyard pictures, mask the cluster, mask the berries, count the berries, and then actually look at cluster closure, so how tight the berries are to one another as a variable that would go into a disease model. And on the lower right is a satellite image from a vineyard with some different sampling schemes. And this is where I'm going to focus my discussion today. But all of the stuff on the right, the digital agriculture, is all to support sustainable vineyard management, right? So we're not trying to make money. We're trying to develop easy to use, free, readily accessible tool that will help growers do what they need to be doing in the vineyard. And so for that, it needs to be so simple, not that they can't adopt anything else, but they're short on time and they don't want to take the time to learn these difficult systems where agriculture has sort of gone in terms of precision agriculture. Excuse me. So we sample vineyards quite a bit, right? Every 3 years or so, we do soil sampling. Once or twice a year, we sample leaf blades and or petioles so that we get a better understanding of nutrient allocations in the vine. And then every year before harvest, we do a significant sampling of fruit composition. So veraison is the start of ripening. And between veraison and harvest, most wineries will sample anywhere from 3 up to about 9 times to check on the maturity of their fruit. So we've always told growers for all of these things, take a large random sample. And the reason we say take a large random sample is that we don't understand the variability in the block, right? So we need to make sure we have a really large sample that's going to encompass that variability so that we don't make a mistake and have an inaccurate sample. But it's hard to take a large random sample, right? It takes a lot of time, takes a lot of money. Even this small vineyard block is like 1.4 acres or something like that. By the time you drive up and down every other row, it is a significant investment of time. And so what happens, growers don't really do it, right? They'll sample from the corners, for example, and then we don't get a very accurate assessment of what it is that we're trying to measure in the vineyard. So maybe about 12 or 13 years ago, when NDVI became more readily available, we realized, wait a second, we have a measure of variability, right? We don't have to take these large random samples because NDVI tells us where there's some variability in the vineyard. And actually, I should take a step back. We originally did some of our sampling work with a manually collected data set. And of course, that went nowhere because nobody wanted to actually go out and manually collect their data set to try to save money on sampling. And so we moved to NDVI. And for those of you that aren't familiar with this index, it's calculated as a ratio between near infrared and red light, and it simply looks at the reflectance of the plants. And so this is just an example of if it's -1, getting closer to 0, very dark. And then the ones that apply to vineyards are on the right-hand side here and tend to be positive. And this allows us to look at where are vines bigger slash more healthy, right? We don't really know what it is about the vine that's driving that signal, but we know bigger vines have a higher NDVI and healthier vines have a higher NDVI. So we started to use this to guide our sample. These are some examples from one of the Chateau Ste. Michelle blocks in Washington State. So on the left, that series of 3 blocks is what comes back when you have an NDVI image. You can set the colors that you want in there. In this example, the dark blue is the most vigorous section, going to the lighter green, and then the brownish is very low vigor, for example, right through here. And so what we do to work with these images is we convert them to black or white, and we can basically go pixel by pixel and get our NDVI values. And then once we have those, we can graph the distribution of what that looks like. So on the x-axis is the value from our NDVI pixel. And on the y-axis is the frequency that we hit that value, right? So it's just a simple distribution. And then our goal is to match that with our sample. So if this is our population over on the left, we want our sample distribution to look very similar. So often they're not going to look exactly similar, but we're going to hit the higher spots and make sure that we're getting there to get our sample. So the goal, if there's one thing to take home out of this whole presentation, is this is the goal so that we can go to fewer spots and do it more cheaper and easily. So the first study that we did, our goal was to determine how efficient a spatially explicit sampling protocol could be. in terms of determining the number of pixels that we needed to sample. So how many pixels in an image do we need to actually sample so that we can get an accurate indication of whatever it is that we're interested in sampling? So to do this, Chateau Ste. Michelle shared with us some drone images that they'd collected with a spatial resolution of 50 centimeters by 50 centimeters. So this was at the time really high spatial resolution. There's better resolution now, but then the question is, what do you do when you get down to 1 centimeter resolution? What do you do with that? So this was 50 by 50, and we used a computer simulation to run a random sample and then to do what we called a globally optimized sample. So that was with a heuristic algorithm called taboo search, which basically goes through the entire image trying to find the appropriate optimization to get to a sample. And I'm going to show you what this looks like. So there are 3 blocks, A1, A2, and A3. You can see our distributions don't look nice and pretty like the previous one did. It's not a normal distribution in these different ones. And then this is the pixel count where we sample for our globally optimized sample in a block. And I'm going to show you a picture in a minute so this makes a little more sense. But I want to highlight these distributions first. So you can see, where it's a little higher, sample. Where it's lower, we sample less, right? And so in this one, it didn't have a sample in the middle because that's considerably lower, but we sample the high parts. And then we also ran a simulation where we told the algorithm, send us up a single row, so we're not driving all around, and give us an optimized sample using a solution that way as well. And so same thing, we look at these distributions. So, and we converted to black and white images, as I said, so I recognize these are not very attractive and it's kind of hard to see. Here is our image across the top. So these are 3 1-acre blocks. And hopefully you can see here, if we turn all the lights off, you can see better, but I won't do that. In this black image, there are white dots, honestly, all over here. And those are the places that we need to go to get our globally optimized sample. In the set of images below, you can see the white dots, and that's where we have to sample just going up a single row in order, again, just to match the pixel distribution, okay? That is the main goal here. And then we're able to quantify what size of a sample we really need. So basically what we do here is we calculate sample fitness. So sample fitness comes from like evolution and then was adopted by computer science. And we're able to calculate this random fitted spline. So this is what we need for a random sample, this line here. And then with our information, we're able to calculate what we need for an optimized sample. And this is just an example. It's going to change every block, okay? Every block is going to have a different version of what we need to assess the variability within that block. But we can calculate how much smaller our sample size can be. So for example, if we wanted to say in a random sample, we were going to sample 20% of the pixels in an image, we can calculate what we would need in our globally optimized sample just by going up, moving over to our spline that's for optimized sampling, which is calculated from the data, come down, and then you can see we only have to sample 10% of the pixels as opposed to 20% of the pixels. But again, this is for this block. This is going to change in every block. So this is what it actually comes out as. So these are 6 different blocks, 3 at site A, 3 at site B. The x-axis here is the optimized sample size, and the y-axis is the percent difference in sample compared to random. Okay, so you'll see our y-axis is negative, and that means it's a drop in the required sample size. And so you can see a lot of our blocks come out we could reduce sample size by about 20%, depending how much of the sample or how much of the population we want to sample. Some come out in the 30s, and then there's one block, just based on the variability in that block or less variability in that block, comes out as a reduction in sample size of more than 60%. And so this is for our globally optimized. This is for the single row. The single row looks basically exactly the same. But here's the beauty of the single row is that you only have to drive up one row as opposed to driving up and down to get your sample through most of your rows or even every other row, right? So then we need to start thinking about saving time and saving money. The other beauty of this smaller sample is it takes less reagents to use in the lab, right? So instead of having a huge sample that requires more reagents, it's considerably smaller. So then started sampling for fruit maturity specifically, okay? So the previous work, we were just looking at pixel values, running mathematical models on them to figure out where we should be sampling. And it was just based on whatever your pixel value was. So we teamed up with Gallo Wines and started working with them on an optimal sampling strategy for fruit maturity. Because if you remember, I said that most growers sample between 3 and 9 times between veraison and harvest. Gallo samples in the 6 to 9 times per season per block. And they're in the Central Valley, so they have blocks that are 150, 200 acres in size for a single vineyard block, right? And how do you sample that? So they had contacted me because they'd seen our earlier work and said, what can we do to work with you to come up with a single sample location? So that's what I'm going to be reporting on here. So we're working in California here, not New York, because we're in these huge blocks that do have less variability than we have in New York vineyards. This was with Landsat 7 because it's what they were using to analyze NDVI for their blocks. Landsat 7 actually just got decommissioned this year. Now we're at Landsat 8 and 9 to get the same values. And we worked with 13 large blocks in year 1 and then 16 large blocks in year 2. So these are the different treatments that we compared. So we always use a random sample. And for that, in all of my studies, we just have the computer pick 20 random spots on the image for us to sample and, excuse me, and give us the location. The next one down is what we call CMAT, or it's also called the Four Corners approach. And I don't know if anybody uses it in vineyards here, but they do use it very commonly in these large California vineyards, which means if you've got a 150-acre vineyard, instead of driving up and down the rows, they go in at the corners, at each of the corners, and take a sample there. And just hope that that represents the variability that's in the block. You can imagine sometimes it does, sometimes it doesn't. And I'm going to show you on the next slide what this actually looks like when we look at the images. And then we used something we derived called NDVI3, where we basically look at the pixel distribution of our NDVI images. We break it into 3 quantiles, and we have our mid-pixel represent the mean of our distribution, and then the next, the pixels on either side represent the tail. And then we tried an NDVI1 solution, which was just one single pixel that is the mean of the image. But this is the last time you'll hear NDVI1 because it didn't work at all. So note that we're sampling considerably fewer pixels as we go down here, but our sample is always the same. It's a total of 20 clusters. So this is what it looks like in reality. So these dotted lines are block outlines. And I see that there's 2 blocks here, but Gallo manages them as a single unit together. Same thing with other ones. The blue dots represent our random sample, again, chosen by the computer to tell us where to go. These peach bars are our CMAT or four corner approach, where you go in and then sample across 2 pixels. And then the red bar is our NDVI3 approach, right, which is based on the pixel distribution in the image. It's just going up one row, crossing 3 pixels, so sampling along 80 meters. So remember, our pixels are 30 meters by 30 meters. So we'd go in 5 meters, sample on either side, and then leave 5 meters at the end of the pixel. So the red line here, the NDVI3, we're just going to one location, walking there and sampling. So we spent a lot of time trying to decide, how are we going to test whether this really works? And luckily, we have some very good statisticians at Cornell who helped us. And we came up with this KS test, which is after two, guessing from the names, Russian scientists, so I'm not going to try and pronounce it. But the KS test basically tests whether distributions are the same. So whether our population and our sample have the same distribution. Okay, again, just working on pixel values here. And so these are the 13 blocks from the first year of the study. The next column over is the total pixels. You can see they're gigantic. If you're at 30 meters by 30 meters and over 400 pixels, that's a very big block. And then here's our KS test against the total population. Okay, the KS test tests random, CMAT, our four corners, and NDVI3. The closer we could get the KS test to 1, it means our sample is a better fit for our total population. Okay, so you can see random does reasonably well. There's a block at the bottom, 0.25, which isn't great, but it passes because it's only considered a fail if it's below 0.05, if the p-value of the test is below 0.05. CMAT, you can see 3 of their 10 blocks fail with that four-corner approach, meaning that the sample distribution didn't match the block distribution. NDVI3, you can see at 12 of the 13 blocks, it performed the best with the highest KS test, and often very close to 1, telling us that we had a really good match to the distribution. But that makes sense, right? Because remember, I told you we started with the distribution, we divided it into 3, our middle pixel was the mean, and then we sampled the 2 tails. So of course, it turned out reasonably well because we derived it from that image. So these are what they look like. So the blocks were all over the place in terms of shape. But here's our NDVI3 solution right in here for this image. Here is our pixel distribution for the entire block in blue. And then here's our NDVI3 distribution. So which pixel values we're sampling across. And so you can see in this one, it really just had a sample directly across and didn't increase numbers at all, because this was not a very significant increase. And then we have these weird shaped blocks. Here's our sample over here. And again, this is for the total sample. This is for our NDVI3. So just to show you where we're sampling in the distribution. So how did this actually look if we take fruit and compare that? So they took fruit from our random sample, 20 clusters, right? And then we did a comparison to NDVI3, our one-stop solution, and CMAT, which is our four-corner solution. And these are basic Pearson correlation coefficients for soluble solids, which is our sugar, titratable acidity, pH, and anthocyanins. And you can see both methods work in terms of representing what's in that random fruit sample. And so these are just examples from year 2 of the study. But when we looked at it over time and in all the different blocks, really we got very good Pearson correlation coefficients. So that told us that our fruit sample, not only looking at pixels, but our actual fruit sample correlated to what we were getting in our random sample as well. So what does this really save us? So these are examples from a recent paper we put out. These are 6 different vineyard blocks, the top 3 in California, the bottom 3 in New York. So the large red star is in the northeast corner of the block. And for this analysis, we always just set the northeast corner of the block as our starting point, for lack of anything else, just to standardize it. The yellow dots are our random sample, okay? And you can see the path that we have to take to get our random sample there. The blue is our CMAT, or 4 corners. And you can again see the path we have to take there. But basically, it's like a box around the vineyard, right? Like we go in a little bit at the corner, but you just kind of continue driving and box the vineyard. And then the red line is our path for NDVI 3. And you can see sometimes it's a short path if the solution ended up being near the northeast corner of the block. Sometimes it's a longer path from the northeast corner, just because that's where the solution ended up being. So a couple of examples. If we look at CA2, so the total is it's 27 hectares, so it's about 68 acres for the block. If you do a random sample in CA2, it takes us a distance of over 13 kilometers, someone driving in the ATV. But if we go to the NDVI 3 solution, it's about 0.6 kilometers. Again, could be much more depending if it was going to be down here. If we look at New York 2, so this is about 5 acres. Our travel distance to get all our yellow samples is about 3.5 kilometers. And it's about 0.6 kilometers to get our NDVI 3. So it saves us a lot of driving in the ATV or walking to do this one-stop solution sample. And this is the cost of a single sampling event. So I had an economics student working on this. And so we calculated labor, fuel for an ATV, amortization of the equipment required, and that sort of thing to come up with these costs. And so for each of these blocks along the top, here's the cost of a single sampling event. So random costs just over $6 in all 3 of our New York blocks. They're all reasonably small. Excuse me. But gets up to about $33 in the largest of the California blocks. The 4C solution ranges up to about $6, again, pretty cheap, but don't forget it failed the KS test in 3 of the 13 blocks. And here's the cost of NDVI 3. NDVI 3 was always under $4 and was a couple of times under $1 in terms of the actual cost of implementing that solution. And then, so if you remember, we said most vineyards sample between 3 and 9 times in a growing season. And so here's our number of sampling events on the left, 3, 6, and 9 for each of these blocks. And so the savings for New York blocks are pretty good, right? Definitely that makes a difference. If a grower has lots of blocks and they're able to save about $50 a block over the season, fantastic. The savings for the larger blocks are even better. So if you remember, I told you Gallo sampled 6 to 9 times. So they were saving $200 to roughly $300 per block by implementing the NDVI 3 method, which they did. We gave them the code, they built their own app, and now this is how they sample all of the Gallo-owned blocks. We're working on getting this method added to My Efficient Vineyard, which I'll show you actually just towards the end, so that growers can use it everywhere. So the next thing we started to sample was leaf nutrients. So this was part of a large SCRI project out of Marcus Keller's lab in Washington State. Everyone was looking at hyperspectral imaging and how we could use that. And we said, we're going to do a new sampling scheme as just a backup in case the hyperspectral doesn't work out. guess what? The hyperspectral still needs a lot of work to get to being able to estimate our macronutrients, but this works reasonably well. So we're trying to look at leaf blade nutrients, but we can't just use NDVI, right? Because I told you NDVI is size of the vine and kind of health of the vine. But what it doesn't account for is soil moisture at all. So in this case, for leaf nutrients, we actually ran with 3 streams of data and then clustered them together. So this is with Sentinel satellites because they've got better resolution. They're run by the European Space Agency. They're 10 meters by 10 meters. So we used Sentinel-2 for NDVI. That was one channel. And then we used Sentinel-1 to get what is called synthetic aperture radar. And there's 2 bands of it, a vertical band and a horizontal band. And they look at backscatter from the soil. And basically that's a proxy for water and soil texture. It's not an actual measurement of it. It's, again, just looking at light reflectance, but it works as a proxy for it. So then we put those 3 things together into clusters. Oh, right. So I wanted to tell you where we got the images, because at Bloom, when we want to sample at Bloom, our canopy isn't big enough to use NDVI because we can't separate it from our cover crops well enough. So for Bloom, for NDVI, we actually use our previous season veraison sample, which seemed kind of out of the blue. And I'll admit, it was a little bit of a grasp, but we've actually calculated that our NDVI images are temporally quite stable over time. And so the differences that exist at veraison often exist at Bloom. It's just harder to work with those images. So at Bloom, we use the previous season, but at veraison, we use that season. So these are what our images look like. This is one of my research vineyards at our farm at Cornell. So the top 2 are SAR, the synthetic aperture radar, the vertical band and the horizontal band. On their own, they don't mean all that much until you put them together. On the left, the green is the NDVI band. And so the darker it is, the more vigorous it is in these corners, and the lighter is the least vigorous. And then we put this together with a technique called K-means clustering, and we come up with 3 clusters in our image. Usually, when we let the solution run, the vast majority of the time, we will come up with 3 clusters. Occasionally, it does come up with 4 clusters. And then, to be honest, we force it to 3 because the 3 works, just not quite as well. And then, I'm not going to get into the whole thing about how we calculate this. Luckily, I had a super smart grad student who's now Dr. Manushi Trivedi, who worked on this. But basically what we do is we take the absolute mean of a pixel value, we compare it, we take the absolute value, we compare it to the pixel mean, okay? Then we put that in the box, and then keep searching around with different 3x3s. I say we, the computer's programmed all to do this, in these 3x3 windows, to find the shortest Euclidean distance, meaning find me a window where we have the least variability from the mean and the shortest walking distance, right? And then it comes up with a box. And so in this case, it says, this is the sample, that's the shortest distance from the mean and a short walking distance. And then we draw a box around it and we sample up actually 3 rows. So here's an example of, again, no, this is the Lansing vineyard. This is one of the grower vineyards in the study. We've got our 3 clusters. And in this example, we did 3 different samplings. Of course, our random 20, the computer picks those locations. They're in yellow. Stratified is just the pixel for each cluster. There's a 3rd one somewhere, or there should be. And then this is our box sample that follows the rows and cuts across 3 of the pixels, or sorry, the 3 clusters, okay? And again, the goal is that it matches the values for the pixel distribution. So when we look at the results, we originally did our analysis with the KS test. Remember where I said we were testing for distributions, but there were too many times it was passing. But we'd look at the distribution and think that as viticulturists doesn't look close enough for us. So then we ended up with the Bhattacharya coefficient, which is somewhat similar, and it maps the overlap of distributions. And so that's what these are here, is these different colors are our distribution. So blue is our random, okay? Red is stratified, and green is our box sample. And so the BC coefficient just tells us how much overlap there is between them or among them. And if the BC coefficient is close to 1, that is better. So these graphs are nitrogen in the top left here, potassium, phosphorus, and magnesium. This is just for one of our blocks, and it's just an example. But you can see in these examples, the BC coefficient was reasonably good. A little lower here for potassium and a little lower over here for phosphorus. And this, I know, looks awful. I'm sorry, we have not been able to come up with a better way of presenting this data. So these are absolute errors, okay, for nitrogen, phosphorus, potassium, magnesium, and calcium. So absolute errors, and then we've coded the color box by the percentage of error compared to the mean. Okay, so basically red is trouble. Red means that our predicted value was more than 10% off of the real value. Okay, yellow is I think 5%, and so there's a gradation. So if we look at nitrogen, basically the worst fail we had was 2 blocks at 5% off. So these are the blocks listed along the left side, and so these first 2 columns are nitrogen at blue, and these are nitrogen at Verizon. So about 5% off, yeah, I could put up with that, right? That's reasonably good in terms of saving the time and effort. Phosphorus, great at blue. Verizon, less so. Potassium, decent at blue. But then we get to these concerning blocks where we're having more trouble getting an accurate estimate. And why is that? And it's because we don't have a yield factor in our model here. And potassium is highly impacted by the yield of the vine as we get to Verizon and beyond. Right? And then you'll see magnesium, not too bad. Calcium, more fails around Verizon. So the take home from this is that the box sampling works great for nitrogen, for all of the macronutrients at Bloom, I should say, and then for nitrogen, phosphorus, and magnesium at Verizon, we're not confident in it when it comes to potassium or calcium. So, but we're excited about this, and I should say these blocks were a mix of New York, California, and Washington state blocks, so they were from all over the country. So in conclusion, spatial sampling can reduce the time and the cost really quite significantly for growers if they can adopt it. The fruit composition sampling, Gallo has an app that's not available to others, but we're having that put into My Efficient Vineyard, which I'm going to show you. Box sampling is in My Efficient Vineyard now and is available for grower use. So the program just downloads the Sentinel images that is required and can calculate the solution for you. So I'm going to switch over and show, oh, I don't know how that came up here. That's super weird. Okay, so this is what I wanted to show you, but I thought I had to go to another tab. So these are some of my my vineyards. Maybe I forgot and added a snip of this. It looks like I did. Okay, so before I show you this, I just want to acknowledge the grad students, Jim Myers, Manusha Tribetti, Kobe Chalk, who did all this excellent work. I'm not the one writing code and figuring this out. My collaborators and funding from USDA and from Gallo Wines. So let me show you. Okay. Efficient Vineyard. How many of you have seen this website before? Okay. 3 people who clearly are interested in viticulture. So I'm showing it to you because it can work for anything. That any like perennial fruit crop that grows in a row type system, it's all the same, right? So this is a platform that's run out of Cornell by Dr. Terry Bates. We're looking to change the name because people think it's only for vineyards, and that's where it started, but it's not actually for vineyards. And so if you go to Efficient Vineyard and go to the login tool over here, you can then mark off your vineyard blocks and start to manage all of your data for your vineyard blocks here. And so I was going to show you, so if we go to plugins here, you can choose satellite image. I can choose the block that I want the satellite image for. And then let's see. So I'm going to pick, it's saying Sentinel-2. This is the Northeast. I got to pick the lowest cloud cover. This is a big problem with working with satellites in New York State. And I'm going to use that selected scene. And then it's going to give me an NDVI image of my block so that I can see pixel to pixel variation. So this is actually, I mean, it doesn't know what it's looking at. This is the barn that's above it. And then, you know, we've got some more vigorous spots along the edges and then a little less so in the middle. And then you can go through here and choose a box sampling. It will go and get the 2 SAR images that's required and then give you a solution. But briefly, I'll tell you before I wind up and answer questions. So we're excited about the box sampling. What we actually think the real application is going to be is in ecological studies. Because imagine you wanted to look at nitrogen that's leaching off of agricultural fields into a nearby waterway. And you're in a large region and you can't go around and sample everything. You can figure out a clustering and go to where that cluster all intersects and do a box sample potentially there. So we actually think the application is not only agriculture, but into ecology as well. And with that, I will wrap up and ask if you've got any questions. Yeah, it's satellite, so it will. So I can't remember because I've had these marked. My plots are marked for a long time, but I think you just go in and you're able to just zoom in and find your spot. There might be a spot where you put in your address. Yeah, but there's lots of cool things here, right? Like it also does. There's a handheld. I'll see if I can find it super quick. There is a handheld data version where like we use it to mark broken posts for the field crew so that then they can just look at where I told them there was a broken post and I don't need to say to them, go to row 14, panel 7, it's loose or something like that. So you can do like a hand data collection with this as well. There's lots of different data layers you can bring in. So I encourage you to try it. There's video tutorials. Those are key. I don't find it comes very naturally how to navigate through it, but I think younger people may find it a little easier to do. But look for the video tutorials if you're struggling at all. Michael.
I had a question. That was fascinating. Excited to see where this is going. And my question goes back to sampling berries, and you have the NDVI. So if you have the app, you're working for Gap, and you're gonna test, go out and sample 6 to 9 times. does, obviously, you know, the canopies are getting larger and the imaging for that block may change temporally. May not be a huge, you may still have the similar patterns throughout the year, but does the app have to recalculate that NDVI tree pathway for that spot every single time? Or is that pretty much a one and done or once a year thing?
They run it as a once a year thing. And we haven't worried about it because we looked at temporal stability and found that we only had enough images of blocks over time, excuse me, to do 3 years. But we found in the Central Valley, where of course the soils are not nearly as variable as in other places, those solutions were temporarily stable for 3 years. So they calculate it every year. I'm confident we've looked at New York images, and I'm confident over a season that it will hold, but I would not risk season to season. I would go in and recalculate every year. But yes, you're sort of one and done for the year. Dr. Schottner. Yeah, so that's a good question. So the question was bare ground cover versus a full-on cover crop, vine to vine, and whether that has an impact. Normally, they'll go through and the students will go through and set a threshold to get rid of the ground cover. We don't have bare. There was bare in the California images, and it just made it easier to basically, this isn't the term they use, but snip those lines out so that you could do it. So yeah, so we try to get rid of the vineyard floor. Although I would argue in New York vineyards, where the vine gets bigger, there's more growth on the vineyard floor. But California is the opposite, right? Their vine gets bigger where there's no growth on the vineyard. So yeah, there are tools to do that. Yes. How does it account for changes in elevation? Like how does NDVI account for changes in elevation? Yeah. I honestly don't know. I mean, it must have a way that it does, but I don't know the answer to that. I'm sorry. Do you have a question of something I know?
Very basic question. They're using NDVI data, satellite DVI, and to help you determine where to go in the vineyard to sample, find out operational fruit.
Yeah.
So there's a issue between greenness of the plant and ripeness of the fruit.
Not really. It just tells us, again, it's just that variability, right? So it's NDVI because it's so easy to get now. And it tells us where there's big vines and smaller vines. But you could do this with a yield map, probably. You could do it with layering a soil map and a yield map. And for juice grapes, that's probably a better option maybe than just straightforward NDVI, right? So the method can use, the statistics are the same regardless of what you use, right? But it's going to have a harder time finding a solution if you're using something that doesn't account for the variability. But NDVI just has a lot of variability.
That variability in the NDVI is because of variability of soil types around the vineyard.
Sure, it can be anything. So, like in my vineyards, it's usually a change in soil type. So you go into a swale, it's got thicker soil, the water's flowing down there, and then we get bigger vines. And then you've got more fruit on the inside that isn't ripening as quickly as where down the row where you've got smaller vines that have better light penetration and the fruits in the sunlight.
That's what I forgot. Exposing taste.
Right. And there's going to be an impact of leaf area to fruit ratio there too, right? And that's going to change bigger vines to smaller vines. And that might also be reflected, but it's all going to kind of come together in the data. So, one thing I'll openly say is that I am very comfortable with our statistics here and they are firm and I believe this works. But there is a lot of hand waving at, yeah, yeah, like NDVI accounts for, shows our variability. There could be other things that show the variability better, but I just don't know what they are or how to get that data set.
When someone Sam, I think I remember, or were you viewing whole clusters?
Whole clusters, yes.
And
Yeah.
how many per vine roughly?
So it was 20 total. So it depended where you were sampling from. If you had 20 locations, you just went and picked one. You went to your location. So Gallo built it so like the app just says like go here and then you just sample there. But if you are only going to one location, then you kind of walk along and take a bunch. Yeah. Yes. Oh, one cluster. Sorry. I mean, yeah, that didn't occur to me. We always sample by cluster, but would it work by grape? I don't know the answer to that because that's introducing more variability, right? Because the front of the cluster is different from the back of the cluster. So if you try it, use the whole cluster. Yeah. Yes. Yeah. Yeah. Grower bias when you're selecting too. Yeah. So we just would literally like, get our cluster that way. At the back. Yeah. Yeah. So we haven't really had any really bad ones. We leave them and just say, okay, we've failed there. And now we don't have a good measure of potassium at Verizon, for example. So it still stays and goes into our analysis. Recalculating isn't going to help. Now, if I added as a fourth layer yield on top of that, then I bet I would do much better. But we're trying to build sort of a straightforward tool for the growers. And so they can't just add in extra layers on top. So yeah, we sort of leave it, but just flag it as this was a fail for us. Let me just see if anyone's on Zoom has any questions. Or if you're on Zoom, do you want to just unmute yourself? Or maybe we're not on Zoom. Okay. Okay. Awesome. Okay. Thank you very much for the